With a co-immunoprecipitation approach, you verify high hypothesis. So you have to have antibodies against the two proteins that you believe that they may interact one with the other one. While in this approach that is called the Yeast-2 hybrid approach, you screen a library of proteins to search for potential unknown interactors, allowing thousands of potential interactions to be screened rapidly. So at first you have the Yeast-2 hybrid system for the screening. You then formulate an hypothesis and you will check with a co-immunoprecipitation or pull-down experiments to verify this hypothesis. But it's very useful for the first screening when you really have no idea of which can be potential interactors with your protein of interest. This technique can be used to discover novel protein partners for your protein of interest. You can also elucidate mechanism of action because you can have a library of modified proteins rather than a general library. You can also find out new functions for uncharacterized proteins, or you can also unravel pathways and molecular machineries. So it's a very useful tool. The Y2H system is based on the reconstitution of a functional transcription factors when two protein or polypeptide of interest interact. Indeed, the eukaryotic transcription factors are modular and consist of sequence specific for the DNA binding domain and another domain that is recruiting the RNA polymerase II. That is a transcription activation domain, or AD. If the DNA binding domain and the activation domain are separated, there is no transcriptional activity. But if those two parts are fused, so they're hybrids with proteins, on one side the protein of interest and on the other side several proteins that may interact, or whole bench of potential interacting proteins. And only when you have the interactions of the two that you reconstruct the transcription factors leading to the transcription of a reporter gene. So here is the wild-type transcription factors. Here we separated the DNA binding domain and the activation domain, and there's no more transcription. Here you have your protein of interest, always as a fusion protein with the DNA binding domain, and a library of constructs in between the activation domain and the different proteins. So only in clones in which you transfected both constructs that interact, you will have a transcription of the reporter gene. So we call the protein of interest here in green the bait, and all the library are the library of the prey. And when there is interaction in between the bait and the specific prey, here we have the transcription. This is basically what happens. So as we said, here is the construct in between the DNA binding domain and the bait, and the library with the prey. When there is transfection of the yeast cells, actually in each cell you have one of the constructs with the bay and one of the constructs with the prey. And you have a selection medium, or you can have also a reporter gene that will give to right to cohort colonies, and those two systems are used to identify the positive colonies. So actually, if we have the interaction, this would lead to a specific phenotype, and usually growth on a selective medium or change in the color of the colony is used. So the most popular reporter genes are the histidine-3, and you grow up your cells in medium without histidine, and drag Z for the screening as a colorimetric assay. So the known protein is fused with the DNA binding domain, and the transcription factor that is used is usually GAL4, and the interacting partner proteins are derived from a library expressed as a fusion protein with the activation domain of the Yeast GAL4 to create the prey proteins. High stringency strains have been developed, for example, by Clontech, using 4 GAL4 responsive promoters, 3 genes for the selection and 1 for the identification. So here we have the different constructs that have been designed, and you can have the different proteins that are different reporters for 3 different GAL4 responsive promoters that are M1, G1, and G2, and they are stably integrated in the genome of a reporter strain. And on the other side, you have a resistance to oxotrophic reporter for histidine or adenine biogenesis that confer growth selection in the presence of Arab on histidine on adenine deficient media. And the last one is the alpha-galactosidase reporter products that will give rise to blue colonies in the presence of X-alpha-gal. So here are some of those vectors. When you have the vectors for the bait and the vectors for the prey, and you can recognize each part of the of those vectors that will confer the DNA binding site. And here is the DNA binding site and the polycloning site with the restriction enzymes, so that you can clone your protein of interest, the cDNA of this protein. And here is the origin of the replication of the vector, and on the other side it gives rise for the antibiotics resistance for the selection of the cloned vectors. We have the vector for the prey, and in that case we will have the sequence that will codify for the activation domain. And again, here we have the multiple cloning site where will be inserted the cDNA sequences of from the library. And this reporter genes will give rise to resistance to ampicillin, so it's antibiotic resistance, and also like Z will produce blue colonies. So here is the results when you have those colonies that are growing as blue colonies due to the lactate and are resistant to the antibiotics. When you get your colonies later on, you have to amplify the positive one. With these techniques, you can have quite a lot of positive and negative results. So it's very important to select all the colonies. You will amplify them, and later on extract the DNA and check where are the different proteins that have been selected, well, the sequence that codify for those colonies, and you will sequence them and identify which is the protein that might interact with your protein of interest. So when you do this experiment, you use an enzyme to digest the wall of the yeast cells, and later on you make the lysis of your yeast cells and purify the plasmid DNA, which can be analyzed directly, or you transform E. coli for the propagation and scale up of the plasmid preparation. And after that, you will go with for PCR amplification and screening and sequencing. You can use this technique, for example, for drug screening, and it's variation that is called 2Yeast+1, meaning that actually you can interfere with the interaction of two proteins of interest in that case. So actually, if we have a situation in which we have A that interacts with B, actually you may screen for molecules that will disrupt this interaction, and you had this third partner, and if there is an interaction in between A, B, and these molecules that you are screening, you may lose your growth and identify in that case with the negative results that your molecule or this specific drug is interacting and inhibiting the interaction. So this is very important because it's a way for a rapid screening for drugs, and since the mean duration of the development of a drug is around 10 years and the associated costs are very high, it has become crucial to streamline drug discovery and development and optimize the drug profiling project from the start. So the understanding of small molecules' mechanism of action through target identification, for example, is now essential to improve drug efficiency and prevent side effects. In this context, chemical biology, in particular chemical proteomics, have been developed and various methods are emerging. So in that case, it's a chemical yeast 2+1 hybrid that is for compound profiling technique derived from the traditional 2-hype technology. And here is the principle of this method, and the detection of an interaction between small molecules, that is here indicated in black, and protein will give rise to the disruption of the interaction, and that can be very easily identified. You can have many variation on applications of this kind, and you can have forward assay, that what we have described right now. You can have forward duplex, so that you will associate two types of constructs, and you have also the reverse assay, in which you will lose the interaction, And in the last one, you have a repressed transactivator. So in that case, you have no growth because the interaction is inhibitory, and if you disturb the interaction, you will get the growth. In any case, you can identify the role of those small molecules. So it's very efficient for screening of small molecules. There are some limits to the yeast to Hybe approach, and one is that, as we said before, you can have positive or negative results pretty easily. So also with the positive results, it's very important to check for by immunoprecipitation technique if there is really a type of interaction. And you can also have negative results. For example, maybe the proteins produced in yeast do not have the right conformation, and the conformation of mammalian protein in yeast is maybe quite different from the normal configuration in the mammalian cells. So it might be important to perform the 2 yeast assay, the 2 hybrid assay in mammalian cells to confirm that the suspected interaction can also take place in the more normal context and environment. So the mammalian assay often reflects interaction between mammalian proteins with greater authenticity than can be achieved in yeast. So the classical 2-Hybe in yeast is not suited for transmembrane protein. It works fine for cytosolic or nuclear proteins interaction, but with transmembranous plasmic membrane proteins interactions, it might not be good enough. So you might use some other approach, and one is called the Split-ubiquitin assay system. And in that case, it takes advantage of the structure of the ubiquitin, and the ubiquitin can be expressed as the N-terminal that is called N-UUB for N-terminal ubiquitin, half of the ubiquitin molecules. And the other one is the C-terminal ubiquitin. And if this one is fused to a reporter gene, for example, you can have, after the interaction in between UUB and CUBE, the recognition of the structure by the protease that will cleave up the ubiquitin and releasing the reporter gene. If there is no interaction, there is no cleavage, and there is a point mutation in the N-terminal ubiquitin half that will interfere with the direct interaction with the C-terminal part of the molecules. So in that case, you don't have spontaneous interaction. You can have the interaction only if you fuse those 2 parts with 2 proteins that will interact one with the other one. So this interaction will bring the 2 parts close enough for the activation or recognition by UBP and the release of the reporter gene. So in that case, you will have the interaction that is mediated by the 2 parts that are the X and the Y protein. So let's see how does it work. Here is the transactivator-based membrane yeast 2-Hybe system. So in that case, you have 2 transmembrane proteins. Here is the protein Y that is fused with the N-terminal mutated ubiquitin, and the X protein that is fused to the C-terminal half of the ubiquitin, and this one is fused with a transcription factor. So if you don't have any interaction, there is no transcription of those repertoire genes. If you have the interactions in between the two, here is the interactions, and we constituted ubiquitin. This one will be cleaved by UBP, and the release of the transcription factors would allow for the expression of the enzyme that will allow the cell to grow in the absence of histidine, and the LAX-D that will give rise to the blue colonies. So we have selecting culture medium and colored colonies. This one is one approach. You can have another one that is called the URA3-based membrane Yeast 2 hybrid system. So the principle at the beginning is more or less the same one. Here is the mutated N-terminal fused with the Y protein, and on the other side we have the X protein fused with the C-terminal part of the ubiquitin, and in that case it's the enzyme that is URA3. In that case, URA3 will be active, and if the medium is containing fly-fluoro-boroacidic, that is called 5-FOA, the URA3 activity will metabolize this toxic 5-FOA, and the cell will die. On the other hand, if there is interaction and the cleavage of the enzyme URA3 means that this enzyme will be rapidly degraded, and the cells will be resistant to 5-FOA. So there's variations of the classical 2-hybrid system that might be very useful for the analysis of the interaction of membrane protein. And thank you for listening to this lecture and the Yeast 2 hybrid technique.